G'day guys, this is the first of a series of videos that I want to make, just covering some of the aspects of the influence of taurine within the gut and sort of the stomach-gut nexus, and to sort of clarify a number of points. So this will be the first one, looking at its interaction with the gut microbiome and its relationship, and then we'll go into the weeds on many other specific points in the following series of videos that will follow this one. Now, let me just share my screen. Anyway, taurine does not affect the composition, diversity, or metabolism of human colonic microbiota simulated in a single batch fermentation system. So there's a Japanese study out of Kobe University, and the Japanese, what they were doing is they were getting some feces samples from a number of volunteers, which to actually see what is the actual interaction between taurine and the gut and stuff like that. So accumulating evidence suggests that dietary taurine exerts beneficial anti-inflammatory effects in the large intestine. In this study, we investigate possible impact of taurine on human colonic microbiota using our single batch fermentation system. Fecal sample from 8 humans were individually cultivated with or without taurine. So in that system, so that's the sort of name of it, Kobe University Human Intestinal Microbiota Model. The results show that taurine remained largely undegraded after 30 hours of culturing in the absence of oxygen. So without any oxygen, basically, it was still around doing its job. But all through summer, 83% of the taurine was degraded after 30 hours of culturing under aerobic conditions, so oxygen. Diversity in bacterial species in the culture was analyzed by 16S RNA gene sequencing. So that's just a method they use as a sequencing machine. You know, nowadays it's all computerized. They can do it within a couple of hours, sequence shitloads of genes, which is very easy to work out. These sort of things years ago used to take a very, very long time, boys and girls. Diversity in bacterial species in the culture was analyzed, revealing that taurine causes no significant change in the diversity of microbiota. Both operational taxonomic unit and Shannon-Weiner index of the cultures were capable. These are just methods which they how they taxonomic is basically, the method of basically, taxonomor comes from a Greek word, which is sort of basically to note, all these different types of entities within the different units. So cultures were comparable to those of the respective source fecal sample. In addition, principal coordinate analysis indicates that taurine did not alter the composition of bacterial species, since the 16S RNA gene profile of bacterial species in the original fecal sample was maintained in each of the cultures with and without taurine. So they were able to see. Furthermore, metabolic analysis revealed that taurine did not affect the composition of short-chain fatty acid production in the culture. These results under these conditions, but artificial conditions, suggest that the beneficial anti-inflammatory dietary effects of dietary taurine in the large intestines are independent of the intestinal microbiota. We infer that the dietary taurine may act directly in the large intestine to exert anti-inflammatory effects. They're quite clearly showing here that, it doesn't change the composition, doesn't alter, basically, it's dietary taurine that's directly doing. When we're talking about taurine, always think in your head, seafood and meat. because that's the primary sources of taurine in our diet, and it's in the fluids of those. So let's make that quite clear. So I'm not going to actually show this, but there's a complex human gut microbiome cultured in an anaerobic intestine on a chip. So they've actually, this group has actually made an actual chip to basically do that. So if somebody wants to see that, I'll add that there. This one is microbiome versus chemistry and the origins of anaerobic gut lumen. This is really important that we realize this, because it'll be important further down. It is generally thought that the gut microbiome consumes oxygen and maintain the lumen in a deep so they believe that it was generally traditionally thought that that's what actually happens in the gut. Bacteria just eat up all the oxygen, okay, and maintain the lumen in a deeply anaerobic state. That means without oxygen. However, we found that the gut of germ-free mice, that means this mammal model, is also deeply anaerobic, suggesting that there exist other mechanisms responsible for oxygen consumption in addition to the microbe respiration. These mechanisms comprise oxidative reactions, but as the oxidation of lipids and other organic substances. Both the microbiota and the oxidative chemistry regulates lumen oxygen levels, and in turn influences the composition of microbial communities throughout the intestinal tract. So there is another mechanism. What other studies have shown is within the actual mucosal layer, and this is important because taurine helps the mucosal layer, protects it, so does vitamin D and stuff like that. That's the location where the oxygen does accumulate to provide for that, which is really important because, by having this sort of good symbiosis and all that, you don't actually end up getting bugs potentially drilling into your gut or stuff like that. So, and as long as they are modulated properly, very important that they don't get out of whack. So this is just to give you a bit of an idea about the microbial life in the digestive tract. These are viable bacteria per gram, and the pH stomach is always much lower because of the acidity levels. Obviously, at 3, our stomachs are 1.5. So basically, there's bugger all bacteria in there. Very few can survive. If you're on a veganurized diet, that's where you get your stomach acidity is going to be, and which basically leaves it open to bacterial colonies in the stomach, which isn't a good thing if you want good digestion. Jejunum is slightly more, obviously. This is in the small intestine. That's the other part of the small intestine, the ileum, and then the colon, which is the large intestine, just so people can get a bit of a perspective. Duodenum, which is this part here. Obviously, this is where the stomach comes in, the contents. This is where, right in here, is where basically your pancreatic ducts come through. Those ones that come in out of the gallbladder, right through the this part, and merge inside basically the pancreas and go right through. That's why if you've got an inflamed gallbladder, you can actually inflame your pancreas as well. And so they mix with the actual enzymes to break down the proteolytic enzymes, and combine to basically within this part to increase the pH and lower the acidity levels through this part. And then you've got this first stage, jejunum, which is where you absorb at a more higher motility most of the stuff. And then in the ileum, which is this other part, is where things are slowed down. They are really slowed down. This is where you do absorb here a bit of water, but not a lot. You do absorb some vitamins and some minerals and a number of other things through this part. But this part is a much slower, the ileum is much slower digestion through this part. This is where most of the stuff spends most of its time. And at this part, you absorb, your B12 in particular. Most of it you absorb at this part. You also absorb your bile salts. You don't want them going into your basically large intestine, causing loose stools, or stuff like that, or, very colored, discolored and stuff like that. You know what I mean? The brown stuff, the light brown stuff, the tanny stuff. And then basically at the end of the ileum, basically most of it actually gets about 90 odd plus gets recycled, as long as everything's working properly in that regard. And then whatever ends up in the large intestines and then out the rectum, basically. So just to give a bit of a perspective for people to get a bit of an idea, let's go down to the other part now. So as you can see, oops. Okay, so that's going to be, oops, that's not going to work. We'll do it this way. As you can see, the aerobic and anaerobic. There's the aerobic, obviously, that's the anaerobic, so hasn't affected at all. Here you can actually see a quite severe reduction in the gut. Taurine's not going to be depleted by opportunistic gut bacteria. But here, obviously, in a petri dish without, it's going to happen. It just shows the difference. So a lot of people, if they're concerned about, I consume taurine, but it's not going to do the job because it's going to be consumed, not going to happen. It's going to be around for quite some time doing a very good job inside it. That's why I always say to people, we set the amount depending on what we're trying to achieve in that regard, just so we're aware. Now let's take a look at the actual, these, as you can see, feces, cultures plus. So that's the general, the control. So there's the culture, there's the culture plus taurine. So you can see not much difference. So it's not basically altering the gut microbiome, which is important because we don't want alterations. We want a healthy gut microbiome, but we don't want, these sort of alterations. So it's not doing it by alteration. So the gut microbiome isn't the thing, because this is the myth that if you have this altered microbiome, you'll actually get less inflammation. It's bullshit. That's what this study is showing, that it's bullshit. In an anaerobic state, basically, taurine is exerting the influence, the anti-inflammatory influence, by a number of mechanisms that I will go through in the following series of stuff. This is the importance, just to basically demonstrate that it's taurine, nothing to do with the gut microbiome, because we've got the vagoons out there making claims which are completely and utterly spurious, absolute nonsense and excrement, So pretty much this is what this whole series, and that just shows you the different strains and all that, pretty much. Now, let's go into this part. I'm going to ignore this first part because I want to minimize the amount of time people can go back and read it. They want, despite its broad distribution in the human body, taurine is not regarded as an essential amino acid. I beg to differ on that, because the compound is not required for protein synthesis. Well, it may not be, well, it is an amino transporter. So it does assist in protein synthesis by bringing in more aminos, but it has other roles beyond that. Nevertheless, taurine has been has a long known to play a range of significant roles in the host physiology and biochemistry, thereby contributing to human health. Taurine, thus, has been increasingly used as one of the most promising dietary supplements capable of providing numerous health benefits. Additionally, several tissue culture-based studies and animal experiments have suggested that taurine has an ameliorating effect against inflammation, bowel disease, involving the human large intestine, which is the colon, including ulcerative colitis, Crohn's disease. However, most, if not all, dietary taurine is normally absorbed in the small intestine, which is equipped with a taurine-specific uptake system, and that's true. It does have an actual system that can actually take up taurine because it's critical for so many tissue throughout the body. But some does get into the large intestine, the colon, and does its magic, because we know that under anaerobic conditions, not going to be eaten by the bugs. That's important. That means it can get in there to do the job, the small amount that gets in there. You don't need massive amounts. Moreover, as a result, it is difficult for taurine to reach the large intestine to exert inflammatory effects. Moreover, we initially assumed that even if some quantity of taurine reached the large intestine, the compound would be readily metabolized by members of the colonic microbiota. That's what was believed. But now, with this experiment, we've shown that they don't need it under anaerobic conditions, only aerobic, only when in the presence of oxygen. And the colon is one of the most anaerobic environments, which is far more likely to survive in there. For example, there's a study showing that taurine was degraded by anaerobic cultures, initiated using cat feces as the inoculum. So, this is just another in vitro study that somebody did with cat feces. We know now from this study that, with oxygen, it's going to happen. So we can pretty much say that all these old studies were confounded with oxygen. So they're irrelevant. However, as we demonstrated in the present study, the majority of the taurine added in the anaerobic, because that's exactly how our digestive system is, in an anaerobic, and they were human bacteria, human feces kept in an anaerobic state. We didn't see that. Exactly. This is not animal stuff. This is human stuff that we're looking at in this model. Cultured initially from each of the 8 human subjects, exactly 8 human subjects, very important. It's human-related microbiome and not basically an animal one, remained intact even after 30 hours incubation. Got it, boys and girls? Furthermore, we found that taurine had no effect on the microbiota composition at the phylum and genus level. So as far as the species goes, no effect. The result contrasts to that report in mice, obviously because they are done in an uncontrolled way, either in vitro or, so that's a problem. Where supplementation with taurine yielded feces with reduced proportion of phylum and proteobacteria. Yeah, we know that because basically, again, it was aerobic. That was the problem. They were poorly done. You've got to look at the design of the studies. Very important, guys, because if you don't look, you miss a lot of things and you make a lot of assumptions. So that's why when I look at these studies, I always look, how were they designed? Were they designed properly to mimic the human gut microbiome, or were they problematic? And a lot of them are problematic. So, we can just discard them immediately. This discrepancy may be ascribed to the differences in microbiome between human and other animals, as demonstrated previously. Yeah, but I suspect there are other, and most of them I actually noticed they have not controlled. They've actually just done feces in petri dishes and not really controlled for the actual aerobic and anaerobic state. So, yeah, I haven't actually really seen controlled aerobic and aerobic conditions. So I suspect that that's the reason, but there could be other factors. That is a potential. They're just throwing it out, but I'm not convinced that much in my personal opinion. Alternatively, the discrepancy may be due to the markedly high dose of dietary taurine. I don't think so. I don't think it's that either in the mouse study. Now, I think actually it was the problem the way that it was actually designed compared to the recommended dose for humans, which is 3 grams for a 70 kilo, which is basically somebody, which is what I take around. I'm doing 4 now, which somebody who is 154 pounds, or this quantity, which they did in the mice, which would be equivalent to nearly 12 grams. So I've used up to therapeutically up to 10, not beyond that point, but still mine is very high, but I don't think it's that reason. That was used in the present study. Recent ROSA demonstrated that taurine supplementation 3 grams significantly reduced, so just 3 grams per day, significantly reduced several markers of inflammation in the human subjects. So they are quite clearly, we've seen, C-reactive protein, interleukins, a whole lot of raft of inflammatory markers, only with 3 grams, go down quite a bit. I usually have just 3 as, 3 or 4 for my immune system and just general stuff. But obviously going higher, I only do that for specific cases and all that. I don't recommend it generally. I don't think physiologically it's normal. You could probably get to that to like a couple of grams if you consumed quite a lot of like oysters and stuff like that. But I don't think you'd get to the 6 to 10, which is sort of therapeutic ones, and they're for specific people who are vastly depleted in taurine and have a whole lot of other conditions. And so they are specific, which I design specific protocols when people email me, and people shouldn't just go around experimenting and playing around without knowing what they're doing. So stick to the 3 to 4, no more, unless you've actually contacted me and we've gone through and I've actually emailed you back and said, well, I think you should be doing this for your specific circumstances. So, but for most people who are generally healthy and just want to sort of improve on that, improve their gut microbiome modularity, sort out their, stomach and gallbladder nexus and stuff like that. In those sort of circumstances, yes, but it's not, it's specific and it should be done in an appropriate way. So do not experiment, stick to the 3 to 4 grams at maximum and no more unless we've had a conversation, because I basically do 3 to 4. Reported that a concentration of, is in vitro. These findings and the results of the study suggest that a trace amount of dietary taurine may escape the absorption of the small intestine in some instances via a drug delivery system designed to carry taurine to the large intestines without being absorbed in the small intestine. No, I just basically believe that higher doses actually have that effect. It's just there is a rate limiting effect how much can it be absorbed in the small intestine. So that's why I usually put people in higher doses when I need to sort things out. Anyway, without being degraded by the human microbiome, in contrast to feline microbiota. Well, we haven't actually proven that in this case. We haven't proven mechanism or anything like that. And the other studies were quite, so I would disagree. And exert anti-inflammatory effects. With that, I agree, because all the studies that I've seen show that quite clearly. Interestingly, we found that most of the supplemental taurine was degraded when cultured aerobically with the human fecal example, and we know that. So only under these circumstances, Orn and colleagues demonstrated that E. coli, one of the common bacteria in the large intestinal microbiota, so our dear friend E. coli that can get to the stomach as well from the, if you're really on a veganerized diet, you can actually have SIBO and actually get our dear friend E. coli all the way up into your stomach. Not a good thing. Imports and utilizes taurine when cultured under aerobic oxygen conditions, implying that the microbial degradation of taurine may require aerobic conditions and only aerobic conditions. In the present study, we utilized cultured vessels and did not consider the effects of taurine on the host immune system. I'll be covering that with other videos on the immune system. I think I've covered it a bit with the T cells and stuff like that, but we'll go into other factors as well. Taurine reportedly activates the intestinal inflammasome to induce downstream generation of antimicrobial peptides, contributing to the construction of a symbiotic intestinal microenvironment. Thus, in the actual human colon, taurine would indirectly affect microbiota composition. And it is exactly in this regard, through inflammasome, that it does. And that will be explained in future videos, and it will all become clear for a lot of people, and the nonsense and the importance of basically taurine in gut health, which basically translate taurine means meat and seafood. That's the best source. It's bugger all in plant foods, unless you basically manufacture it artificially in a lab. Pretty much it comes from animal foods, and that is why the superiority of animal foods in basically maintaining healthy, balanced gut microbiome, and also a direct anti-inflammatory effect through inflammasome. And I will explain that in future videos. This was just to do the groundwork to clear up a lot of confused information that exists on the functioning of the gut microbiome in the general sense. Anyway, see yous.